We had some technical difficulties Tuesday, so my screen flow, my capture thing wasn't compatible with Sierra, so I had to get an upgrade. So we should be back in business. Do you guys watch those videos, or what do you think? Oh, you do? Okay. Okay. Do you watch them on YouTube or Moodle? Moodle. Is it easier? Okay. Okay. So kind of use those, I mean, I'd use those to help you understand material, get clarification, and, those sorts of things. So when's the exam? Okay, you want it easy or hard? Easy. Really? Yes. Oh, I always make the first one really hard to kind of weed people out, to make sure. Okay. When's the draw? Yeah. So, well, it'll be pretty similar. Yeah. Yeah. Green one. Yep. So you'll need a green Scantron. So that, yeah, it should be. That's on the study. That should all be on the study guide, I think. So yeah, bring a green Scantron, number 2 pencil, and a calculator. Okay. So you know what kind of work problems are going to be on there, and then there are going to be 2 review sessions. One tomorrow at 9:30 in here, and then another one on Monday at 2. Is that what I said? 2 o'clock. Okay. So I'll record each one, put it on Moodle and YouTube so you can watch it, and so hopefully. So when you come to those review sessions, just don't come, oh, Dr. Johnson, what's the answer to this question? Don't come with those sorts of questions. Just give me some idea that you kind of, you've looked at this question, you looked at the material associated with it, maybe from the study guide, and you're looking at a graph and you're saying, I don't really understand the point of this or what the data show here, some sort of directed questions. So I'm not going to come in and just lecture and start going through the study guide. I'll just entertain questions from students. So I know that's sometimes a little difficult, but it's a group session, so, okay. And I know some of you may not be able to make any of those times, but, yes. Yep, and I have office hours, so you can email me, if you do kind of have, short directed questions that I can answer pretty easily, I'd be happy to do that. Or you can come, I have office hours on tomorrow. Well, no, anyway, you'll look in the syllabus. I have it on my calendar. I think it's Monday I have them in the morning, I think. Anyway. But if you can't come to any of those things and you don't want to email me, you can try to just tell me you want to come by, and I'll try to set up times to meet with you, okay? Anything else? Oh, yeah, the inquisitive stuff, right? If you want possibly, what would I say, 4 extra credit points if you get 75% on each module, you get 2 per each. So try to finish those if you can. I extended it beyond Tuesday, so Wednesday, so if you can do those, and if you're having any trouble getting into that. So they're kind of difficult, but I think I think it's not too hard to get above 75%. But. Nah, I mean, it's all from the same publisher. They're different, right? But I think all of the things that you see on Moodle, those practice quizzes, the test bank that I have, and those things are all developed by the same person. So I think they're kind of similar. But there are a lot of things on those modules that I've never touched on. So I understand that can be a challenge. Okay. Any questions about the exam or other things? Okay. What we're going to do today is we're going to finish up looking at evolution in finite populations. And the final assumption of Hardy-Weinberg we're going to look at is the idea that populations are infinite in size. And there's a reason we might be interested in this, right? Can you tell me why we might be interested in the effects of perhaps small population sizes on genetic variation? What? Yeah, that assumption, right, that there are no infinite populations. There are some bacterial populations, viral populations that obviously are extremely large. But in general, most organisms, and especially vertebrates, have fairly limited population sizes. And one of the things we're really interested in is especially certain populations of organisms are losing population size. And we'll talk about various things that are causing reductions in population size, but we're very concerned about the effects of decreasing population size on genetic diversity and the effects of organisms to deal with that. So that's kind of one of the reasons we want to look at this. So let me just, I read the book today. I don't believe everything that's in a book. So this one I'm still puzzled about. So Here's a cat. I love the one on the bottom. I think that's a caricature, but I think it's trying to exaggerate the phenotype of this particular cat. But one of the things, there's this Manx cat that's found on the Isle of Man, which is an island between Ireland and Great Britain. And you find this cat, and it's still there. And it was believed to have been introduced by, it's believed by Spaniards maybe 400 to 500 years ago. And that's what it is. What's weird about it? The legs are extremely elongated, relative, and what else is missing? Yeah, it looks like my feral cat that I take care of. Okay, except for the legs. Okay, so it's an unusual phenotype, and it turns out this is a fairly simple mutation that results in that phenotype. And it's called the Manx phenotype, and it's only the heterozygotes that have that weird, loss of tail and elongated limbs. And MM are normal cats. Turns out this is an extremely deleterious gene copy, especially when it's in big M homozygotes. Basically, all individuals die. It's believed to occur when it occurs before they're even born. It's during embryo, during gestation. All the individuals that are big M die. So that's an extreme. Would you consider that a pretty deleterious gene? That's about as extreme as you can get, right? It's basically a lethal gene when it's in combination, when they're both copies of the MM gene. So the question is, how is this maintained on the Isle of Man? What do you think? What do you think the outcome should be now that you know something about selection? What do you think should happen if just selection were occurring? Yeah, you would think that eventually what should happen here? You should get the fixation of what particular gene copy or allele. You'd think the small m, right? So what the book says, and they kind of leave you hanging and they never come back to it, is they say, oh, this is due to what's called a founder effect. That basically what happened is the Spaniards brought this phenotype in, a very limited number of individuals, maybe 2 to 5, and an extremely small limited population size. And that's how that deleterious allele is maintained. I find that hard to believe. But anyway, I'm not going to ask you those sorts of questions, because I would think selection would overwhelm that and get rid of that gene very quickly. I suspect what's going on is, I think people on the island, guess what? I think they like those cats. Anyway, I think there's some sort of selective breeding that's going on, but I don't know all the details. Anyway, so they say this has to do something with what's called founder effect, which is where populations are founded by a very small number of individuals. And we're going to be looking at what's called founder effect, and then we're going to be looking at another thing that they're called bottlenecks. And we're going to be looking at the effects of small population sizes on genetic variation, both within and between populations. So we're going to kind of keep with the same thing. So here are the kind of basic questions. How does the process of evolution in small populations differ from that seen in large or infinitely large populations? How does genetic drift work, and what are its consequences? And the consequences we're going to look at is what happens to the genetic variability within and among populations that are experiencing small population sizes and genetic drift. And then we're going to kind of look at two kinds of genetic drift mechanisms. One are called population bottlenecks, and the other one are founder effects. How do they contribute to evolutionary change? And then at the end, we'll look very quickly at how genetic drift can interact with selection. So again, kind of the same themes. So, you haven't done this. You haven't done the drift example. You haven't done the drift in lab yet. But so you'll be doing some experiments with drift that I hope will illustrate this. So the way we have to think about this is when we think about small population size and compare that to infinitely large populations, what you get are what are called random fluctuations in gene frequencies. They're often called stochastic. They're unpredictable or stochastic changes in allele frequencies over generations and over time. And it's due to what's called sampling error in small populations. And we're going to try to illustrate it. And I think it's easy to understand if you were to flip a coin 10 times, right? If you think about it, if you were to flip a coin, what do you think is the most probable outcome in terms of the number of heads and tails during those 10 flips, in terms of the total number of each? Yeah, in general, you would think, probably your highest outcome if you did that, if 200 people did that 10, those flips 10 times, your biggest outcome is going to be 5 heads and 5 tails. Do you think that's always true, though? What you're going to find when you have really limited population size, you're going to find some cases where you're going to get 3 heads and 7 tails, or 7 heads and 3 tails, right? If you do that, if you were to flip that coin 100,000 times or 1,000,000 times, what do you think of that distribution around 50-50 is going to be? Do you think it's going to be as wide as when you do 10 individuals, whereas if you flipped it 1,000,000 times? Turns out you don't want to do that exercise, but it can be done very easily through simulation, right? And what you're going to get is you're going to get, in those cases, almost exactly all of the numerous times you do that. If you do 100 times, most of them are going to be 500,000 heads, 500,000 tails. You on occasion are going to get 490, but a very small deviation around that 50-50. Whereas in small populations, you're going to get this huge, a broader distribution of things. So that's kind of what it is. So let's kind of go through this example. In this example, there are 10 parents in a population, okay? There are their genotypes, and the frequency of the A1 allele is 0.5. So if you determine all that, and you can figure out 0.5. What we're assuming here is these 10 parents are then spewing their gametes into a large gamete pool. So you're producing a fair number of large gamete pools. And the allele frequencies in the gamete pool are exactly reflect the allele frequencies in the parental population. So they're 50% are A2 and 50% are A1. So we haven't seen any what are called deviations from those starting frequencies. It's exactly the same. Now we're assuming we only are drawing a few number of gametes at random from the gamete pool. Instead of all of these pairing together, or if you had 100,000 or 1,000,000 in there, you're only allowing 10 zygotes to be formed. Okay? So draw 2 end gametes at random from the gamete pool. So just go in and don't look at it and pick them out at random. Pick 2 things out at random, they become a zygote, do it again, and you do that 10 times. If you just pick out those things at random without looking at them, right? What you're going to get by random chance is allele frequencies in the sample may not be exactly the same as the allele frequencies in the gamete pool. And it turns out, you can figure out all this, but it turns out the gamete pairs will form N offspring, and here the frequency of the A1 allele has, by chance, increased from 0.5. It's become much more common. It's become 0.75, okay? And this is just considered kind of a random stochastic effect that occurs when in small populations. And this is a very common phenomenon in nature. So it's this process where you have a small number of gamete pairs in a very limited population size that can cause a deviation. Is it in Hardy-Weinberg? Is it a non-changing frequency? No, we violated the assumption of Hardy-Weinberg, which is infinite population size. So in small populations, you can get these sort of stochastic random changes in allele frequencies. Okay. Did biodiversity talk about this at all? Do you remember this? You don't remember it? Okay. Most people avoid it. I don't know why. Okay. It's a little kind of conceptual. It's kind of difficult to understand, but hopefully. So what I'm really interested in is what happens, right? What happens to allele frequency changes over time, and what happens to genetic variability within populations under varying levels of population size? So these are 3 simulations. You'll be doing these in lab in next week, I believe. And you're probably thinking to yourself, why didn't I do this before the first exam? Okay. So here in the top are a series of simulations where population size is 10. Here's a little larger population size, 100, and here's 1,000. None of these, are any of these infinitely large? No. They're not in the millions or billions. These are still small, relatively small population sizes. So what we do here, we start out at time 0. Along the y-axis is the frequency of the A1 allele. In the opposite way, you can look at the mirror of that is going to be the frequency of the A2 allele. Okay. So let's look at this. Each line in this, they all start out at the starting allele frequency at 0.5. So each one of them, 0.5 and 0.5. There's the starting allele frequency of A1, equal frequencies of both. Each line represents an independent population. So this would be a case, I think, where they did 10 simulations and replicated the same thing, given a population size of 10, 100, and 1,000. What strikes you is the most, what strikes you about these graphs? Okay. So, yeah, so what you'll notice here is this is very different from this. And what you'll notice is, what about, when you look at this, what happens in this small population size at 10? So when you think about this, here's one generation. It randomly goes down, right? If we just kind of watch this blue line, oh, then it randomly goes up. Then it goes down, then it goes up, and then it goes up, and then it goes down. And there's this sort of stochastic effect. But as you start reaching points up in here, what happens? You'll notice in small population size, you either go to, what happened to all these, in 4 out of the 10 populations, what happened? Fixation, right? Of which allele? The A1 allele. What happened in, I can't see all these lines, 1, 2, 3, 4, 5, I think they're 5. Maybe they're 5, and who knows? I can't count them. Maybe they're 8, who knows? What happened in the other populations? They lost the A1 allele and went to fixation of the A2 allele, okay? Did that happen here? So here what you can see in a larger population size, are you still seeing sort of stochastic random changes each generation? Yeah. But the magnitude of those oscillations, right, they don't go to fixation. What you do find is, did the populations diverge from one another? They all started at the same frequency. Yeah, you can see they did. Some went to 0.3, right, for the A1 allele. Others went to about 0.6. So they became a little different from one another, but not to the extent that you see in a really small population. Okay? And here, what's this one? It's kind of in between, right? So what you can see here, the key take-home message is under extremely small population size, what does it lead to? Fixation. What happens to genetic variation within populations? It's lost, right? What happens to the differences between populations? It becomes pretty dramatic, doesn't it? Yeah. You can see they fix for different alleles, and the allele frequencies become extremely different. And so it leads to these populations are fixed for the A2 allele. These populations are fixed for the A1 allele. So genetic drift, in general, you lose variation within populations, but it also leads to divergence between populations, especially at really small population sizes. What would, if you did this simulation with a population size of 10 million, what do you think those 10 lines would look like? They would basically be, they would probably be indistinguishable from one another. They would all be very closely centered around 0.5. They would not oscillate or show those random changes, okay? And you may do that. I don't think they make you do, because it's too much computational time on those Macs. So, but if you did that, you'd basically find 10 lines that were all right on top of one another, right around 0.5. And that would kind of indicate what? Is genetic drift operating in a population of that size? Probably not, or it's so minimal it's not important. Okay? So what are the effects? This is, I think this is New Zealand snapper, I think. I can't remember. I forgot to look in the book. One of the consequences, and I'm not going to go into what the book goes into great detail about how to calculate expected heterozygosity and all these things under genetic drift, and we're not worried too much about that. So one of the areas of concern is populations, especially in fisheries, can become reduced due to overfishing. And one of the things that people are really interested in is sort of developing what are called sustainable fisheries, right? Such that, obviously we want to harvest them, right? They may be wild, harvestable populations. but we may want to harvest them at a rate that doesn't cause dramatic declines in population size. So a lot of people are interested in designating and determining methodologies to determine ratings in terms of fisheries sustainability. And obviously the Gulf region is actually in the midst of this right now, and are starting to develop some measures and indices of sustainability of various kinds of fisheries, from just fish to crab to shrimp to all sorts of things. So it's something that is of interest to a lot of people here, even locally. So again, this isn't a dramatic decline, but here's data over a number of years. They actually could go in and had preserved specimens of these things. They could go in and determine at genes, the sort of heterozygosity. And there was a series, I think it occurred in the 80s, of extreme overharvesting of these, and this actually happened in Louisiana too. Do you know what that was associated with? The blackened Paul Prudhomme. Oh yeah, blackened redfish. The craze started. It actually resulted in kind of an overfished redfish population. It's recovered very quickly, but anyway. So what they showed here is what happened is over time, this was I can't remember exactly when the overfishing occurred in this New Zealand fishery, but what they showed is there was a decline in heterozygosity or genetic variability, and presumably due to overfishing. Sometimes that's difficult to determine whether it's due to one thing or the other, and it often can be very complicated, but at least in the book here they say that this decline in genetic variability as measured by heterozygosity was attributable to overfishing. So this is a concern for people. So this kind of illustrates in a very complicated way the process of genetic drift, and I'm not going to worry too much about this. But what I'm going to try to show you is we're going to start out, here's time 0, and we'll go through 32 generations or years or whatever it is. And what we're really looking at is the outcome, how we have 5 island populations here, right? They each start at this population. Here are copies of the A1 allele. So they're basically equal copies of the A1 and A2 allele. And if you go in and count these, it's 50% A2, 50% A1 in each of these 5 populations. If you impose, assuming there's genetic drift, these are pretty small population sizes, not many individuals. Genetic drift, the process that occurs, and over time, what you can see is, we'll just go down here kind of towards the end. So these are the percentage of these populations that now have 0 copies of the A1 allele or 20 copies. So there are 10 individuals, 20 copies of the A2 allele. And there are a few instances where some populations have copies of both A1 and A2. But what do you notice happens over time? We don't need to worry about all the details and the process that's occurring here, but what it's showing is drift is occurring. This is the final outcome. What's happened here? Yeah, you've kind of gone within each population. What's happened? Think about that graph I just showed previously. These 2 populations, what happened to them? They went to fixation for the A1 allele. So I think they're 2 divided by 5 should be 40%, okay? So these are the copies of the allele. So all of these 2 populations went to fixation of the A1 allele. What happened to these 2 populations? They went to fixation for the A2 allele. What about this one? Has it gone to fixation? You think it will soon? Yeah. So this is kind of what was shown in the previous graphic, but kind of in a different way, is that this whole process of really small population size, genetic drift leads to fixation of genes or alleles within populations, but drift leads to genetic differentiation between these island populations. So these are very different from these and those sorts of things. So this is, if you can remember anything about genetic drift, this is probably one of the most important concepts. Okay, let's kind of put an example on this of how small population sizes or larger population sizes affect genetic variation. These are Galapagos lizards. This is off the coast of Ecuador, right? Yep, fairly far out in the ocean. And This is a dynamic place, lots of interesting animals. You all remember Darwin's finches, right? Okay. So this is a snapshot. We're just looking at these islands right in here. So sea level is a dynamic thing, right? And I suspect it's going to become even more dynamic, right? So let's look at kind of the history of sea levels. And what we're going to be looking at is how sea level has affected the amount of the island area and the connection between islands and these sorts of things. So what's represented in this color is, this is the outline of Isla Santa Cruz, and this is the current sea level, okay? Out here is the sea level about 12,000 years ago, and there's the sea level about 17,000 years ago. What happened 17 to 12,000 years ago, and what effect did that have on the sea level? Huh? Yeah, land bridge. Well, actually what happened in the late Pleistocene, 12 to 17,000 years ago? There were a lot of, was it cooler or warmer than it is now? It was considerably cooler, and it was an ice age. And there was a lot of water tied up into glaciers in polar regions, okay? So a lot of that water was tied up in glaciers, right? So what do you think happened to sea levels during that time? It dropped, okay? So sea levels were much lower during the late Pleistocene, during the last ice age. And this was about 12 to 17,000 years ago. So so what happened? So when you think about that, what effect did that have on the size of populations and the connection between what now look like isolated islands? Well, it turns out these populations were much bigger at that time. They had not this smaller area, but this much larger area. Santa Fe, you look down here, here's its current size. This is where it's this is its projected size, 17,000 years ago, okay? So they were much larger in general. So what's happened over time? What's happened to the island areas? They've shrunk, right? What's happened to the connection between populations? Has the connection become greater or less? Have they become more isolated or less isolated? They've become more isolated, right? So you can't really see it here, and it's mostly in these areas, right? That, it turns out a lot of these islands were actually, all of these things were actually connected and part of the land of that island at the time. And then as the sea levels rose, right, they became isolated. So what's happened is islands have become smaller, and these islands have become isolated from the bigger islands, okay? So this is what's happened. What's been the consequence? And I'm not going to get too much in terms of and I'm not going to worry, these are all based on genes called microsatellites, and your book goes into great detail about that, but there are lots of different ways you can measure diversity of alleles in populations. This was done through what are called tandem repeats or microsatellites. And what would be your take-home message from this? So which one's Isla Santa Cruz, do you think? It's probably that one. I think it's the biggest island. Here's some of the smaller islands. Which one has greater genetic diversity as measured by the mean number of alleles? What do you see? What's the pattern here? Smaller islands, right, have lower genetic diversity. Larger islands have greater genetic diversity. This doesn't necessarily mean that drift isn't occurring here, but drift is much more pronounced. The loss of genetic variation in really small populations is much more pronounced on these smaller islands. So a lot of these Galapagos lizard island populations on these really small remnant islands have extremely limited genetic variability. Okay. Should we be concerned about that? I would be. I don't know if you are, but I would be. But we might be worried that this limited genetic diversity might limit their responses to some sort of changes. It could be how do they deal with diseases, how do they deal with all sorts of things, environmental variability. Maybe they're less able to cope with environmental changes. That's a possibility, and that's something we might want to look at. Okay. Okay, let's do an eye clicker. So I realize this is really hard to see. This came from the test bank, okay? So the top one is graph A, right, with 10 individuals. The one in the middle is what? 100, okay? So the questions are, let me. Okay, so the answers are there is no difference between the populations. The differences in the graphs are due to chance. The populations in the graph on top have a longer generation time than the populations in the bottom graph. The populations in the graph on top are experiencing genetic drift. The populations in the graph on bottom are experiencing directional selection. And D is the population sizes in the graph on top are smaller than the population sizes in the graph on the bottom. I guess I gave away the answer, didn't I? Oh, okay. Okay, choose. This is kind of weird because I looked at this graph and I said, boy, this is really easy. And the test bank said this is moderate hardness. So you know where I come down on difficulty. Okay. But these are the kinds of, I'll pull some questions from the test bank that are similar to this. So I'll even pull one or two maybe from the quizzes on Moodle, if I'm in a good mood. Okay, everybody in? Okay, what's the answer? Okay. So C, some people chose C. This really doesn't have necessarily anything to do with directional selection, right? So this is mostly just looking at drift and how the effects of population sizes are much more pronounced in a population with smaller sizes. Okay. We'll do another one towards the end. Okay. Let me stop this.